Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 9th December 2020. As you all know that we are into the revision mode, we are revising very important chapter called respiration in plants, chapter number 14, and in this session we have been talking about reflecting upon and appreciating the food that we eat finally gives us a lot of energy. And whatever food that we eat in the form of breakfast, lunch, dinner, snacks, tea, coffee, and whatever liquids we take, all will be finally the end product of digestion is glucose. And once glucose is entering inside each and every cell, it combines with oxygen and gets oxidized. So we say oxidation of glucose. So respiration technically we say is the oxidation of glucose. And when glucose molecule starts breaking down in phased manner, it releases a lot of energy. As a part of it, the first step of breaking is glycolysis. Glucose lysis breaking. So in our previous session, we talked a lot and reflected upon, appreciated the all important step called glycolysis. And at the end of the glycolysis, all of us appreciate because it gives us 8 molecules of ATP. And further, when this broken glucose having 2 pyruvate molecules, now these 2 pyruvate molecules enter into next phase, step number 2, that is Krebs cycle. And of course, as the name suggests, we must be thankful to the greatest scientist, Krebs, Hans Adolf Krebs, in 1937, way back. So it is also called by his name as Krebs cycle, and technically it is called TCA cycle, tricarboxylic acid cycle. So the citric acid cycle, other name for this also, was proposed by Hans Adolf Krebs in 1937 based on the studies of oxygen consumption in pigeon breast muscle. Krebs cycle operates in mitochondrial matrix. You remember the powerhouse of the cell, because it contains a complex mixture of various respiratory enzymes, whereas glycolysis takes place in cytoplasm. Here it is in the matrix of the mitochondria. Acetyl-CoA, you remember, 2-carbon compound, combines with oxaloacetate, that's a 4-carbon compound. See, oxaloacetate here, 4-carbon, acetyl-CoA, 2-carbon. So 4 plus 2, 6-carbon. That 6-carbon forms citric acid. That's why it is called citric acid cycle. So a 2-carbon acetyl-CoA combines with 4-carbon oxaloacetate in the presence of condensing enzyme called citrate synthase to form a tricarboxylic 6-carbon compound called citric acid. This is the first step in Krebs cycle. And this citric acid also called as citrate. It is the first product of Krebs cycle, and CoA, that is coenzyme, is liberated. Now citric acid or citrate undergoes reorganization in the presence of aconitase, forming cis-aconitate. Look at that, cis-aconitate, and water. So dehydration, water will be out. Cis-aconitate is converted to isocitrate. Again, all these are 4 plus 2, 6-carbon ones. Isocitrate with addition of water in the presence of an enzyme called aconitase. Since water is added, it is hydration. Whenever water is removed, remember, dehydration. Whenever water is added, it is hydration. Isocitrate now is dehydrogenated. Isocitrate is dehydrogenated, removal of hydrogen, to form oxalosuccinic acid or oxalosuccinate in the presence of an enzyme isocitrate dehydrogenase. And of course, manganese Mn2 plus is a must. And what is important, the most energetic compound NADH2 is produced, and this is referred as dehydrogenation 1. And oxalosuccinate is decarboxylated, means carbon is removed, CO2 minus, carbon dioxide is removed, decarboxylated to form alpha ketoglutaric acid. Since one carbon is removed from 6 carbon oxalosuccinate, when alpha ketoglutaric acid formed, since 1 minus C is minus, it becomes 5 carbon compound. So alpha ketoglutarate will be formed in the presence of an enzyme called decarboxylase. This step releases one CO2 molecule, hence we can say decarboxylation 1. Alpha ketoglutaric acid is both dehydrogenated, hydrogen is removed with the help of NAD plus, and decarboxylated, carbon also removed, by an enzyme complex called alpha ketoglutarate dehydrogenase. So this is dehydrogenation step number 2 as well as decarboxylation step number 2. So formation of alpha ketoglutarate. The product combines with CoA, okay, and it forms what is called succinyl CoA. So 5 carbon compound alpha ketoglutaric acid is becoming 4 carbon succinyl CoA. To continue the cycle, succinyl CoA is converted to succinic acid, which is again 4 carbon, by the enzyme succinyl thiokinase, or it is also called succinyl CoA synthetase. This reaction requires GDP. Look at that, GDP. That is, you may be remembering, I said GDP is guanosine diphosphate. GDP, A, adenine, G, guanosine or guanine. In the presence of inorganic phosphate, the high energy bond of succinyl CoA is transferred to GDP, converting it to GTP. Diphosphate is becoming triphosphate. And this is the only step in the citric acid cycle where a high energy phosphate bond is generated at the substrate level. So since GD di is becoming GT tri, one phosphorus added, so phosphorylation by the action of phosphokinase. ATP may be formed from GDP because both are equal. A nucleoside diphosphockinase catalyzes phosphate transfer from GTP to ADP. Next, succinic acid is metabolized by fumaric acid by dehydrogenation, catalyzed by succinic dehydrogenase. It is the only dehydrogenation reaction in the citric acid cycle that involves direct transfer of hydrogen from the substrate to FAD without participation of NAD. That is how fumarate or fumaric acid is formed. A molecule of water gets added to fumarate or to fumaric acid to form 4 carbon malic acid, and here you can see FAD is getting converted to FADH2, very high energetic one again. So the molecule of water gets added to fumaric acid to form malate or malic acid. The enzyme is called fumarase. So since hydration is taking place, this is second hydration. Malic acid is dehydrogenated or oxidized through the agency of malate dehydrogenase to produce oxaloacetic acid. You can see the formation, NAD becoming NADH plus H plus. That's why hydrogenation. And The oxaloacetic acid is again a four carbon compound. Hydrogen is accepted by NAD plus, that is dehydration fourth. Oxaloacetate picks up another molecule of activated acetate to repeat the cycle. Once this is formed, again it picks up another acetyl coenzyme and one more cycle. So as two molecules of acetyl CoA are formed from one molecule of glucose, remember during glycolysis, from one molecule of glucose, two molecules of acetyl CoA are formed. Now from one molecule of glucose, a total of 24 ATPs are produced from one acetyl CoA. Net equation of Krebs cycle is, we can write 2 acetyl CoA, let me write here, 2 acetyl CoA, 2 acetyl CoA plus 6 water molecules plus 6 NAD plus and 2 FAD, 2 GDP, guanine diphosphate, and 2 inorganic phosphate. So what do they become? They become 2 acetyl become 4 carbon dioxide, 4 CO2, okay, 4 CO2 plus 2 molecules of water will be released, plus 6 NADH molecules are formed, 6 NADH, and of course 6 H plus, and then 2 FAD is there, no? So that becomes 2 FADH2, FADH plus H plus, and 2 GDP will become 2 GTP, guanine triphosphate. And of course at the end of the cycle you are getting 2 coenzymes, coenzyme A. So being catabolic, because it's breaking, what is being broken? The pyruvic acid. So being catabolic, Krebs cycle provides a number of intermediates which are used in different anabolic pathways to form important biomolecules like glutamic acid, aspartic acid. Hence this cycle is amphibolic. Amphibia means both the ways. Here amphibolic in nature. Amphibolic pathway is the one which is used for both catabolism as well as anabolism. Catabolism, breaking down. Anabolism, making or building up reactions. Hence, thanks to Krebs cycle, it is amphibolic in nature. Now, I want you to practice this entire cycle because this is a major question. This is a 5-mark question. Hence, first thing you need to understand. Don't get scared because of so many names of chemicals. Why you need to appreciate this is in every cell in our body, this is going on every minute. Hence, we need to understand what is going on inside. We need to appreciate how we are getting energy, and who doesn't want energy? Everyone wants to be energized. And to know the secret of getting energy is to understand Krebs cycle, to understand glycolysis. So keep practicing, not just for exam's sake.